Welcome everybody. We are discussing the chapter Cell, the Unit of Life. This is chapter 8 of class 11th Biology. In our previous two sessions, we have discussed Introduction to Cell, Prokaryotic Cell, Eukaryotic Cell, and in Eukaryotic Cell we have already discussed cell membrane and cell wall. Today in this session, in the third session, we are going to discuss the cytoplasm, and in the cytoplasm the cell inclusion and cell organelles are there, and the cell organelles are divided into two categories. One category constitutes the endomembrane system. So today we are going to discuss the endomembrane system of the cytoplasm. So let us start our discussion. So cytoplasm, what is cytoplasm? Cytoplasm is the part of cell that is present between the nucleus and the plasma membrane, which contains various organelles and inclusions. Okay, and this cytoplasm constitutes about 90% of the 90% of the cytoplasm is water. So it is a semi-fluid structure, we can say, and besides water it contains the ions, different ions, biomolecules like sugar, amino acids, nucleotides, RNA, enzymes, vitamins, etc. And these biomolecules are known as the cell inclusions, and besides this, the cell organelles or the cytoplasmic organelles, which perform very specific function, are also present. They are plastid, lysosome, spherosome, peroxisome, glyoxysome, mitochondria, ribosome, centrosome, flagella, cilia, etc. So here in this picture, I am going to show you. So this is the cell membrane or the plasma membrane, and this is the nucleus, and in between the nucleus and the cell membrane, the part which is present is called cytoplasm. And here in this cytoplasm, you can see that there are several structures are present, and these structures are the cell organelles. Okay. So now, in the cell organelles are of two types. One is a group of organelle that constitutes the endomembrane system. So what is this endomembrane system? These are the membrane bound organelles which work in a coordinated way. So these organelles are endoplasmic reticulum, Golgi complex, lysosome and vacuoles. So these organelles are working in a coordinated manner. So their functions are related to each other. So they are known as endomembrane system. And besides this, the organelles like mitochondria, chloroplast, peroxisome are not coordinated with the above organelles like endoplasmic reticulum, Golgi complex, lysosome and vacuoles. So they are not considered as part of the endomembrane system. So let us discuss the organelles of endomembrane system one by one. The first organelle is endoplasmic reticulum. The short form is ER. So what is endoplasmic reticulum? It is a network or reticulum of tiny tubular structures scattered in the cytoplasm, and it is present in almost all the eukaryotic cells, but it is absent in ova, I mean egg, embryonic cells, mature RBC, and it is also absent in the prokaryotic cells. And these endoplasmic reticulums are either absent or poorly developed in rapidly dividing cells. And regarding the structure of endoplasmic reticulum, the endoplasmic reticulum is made up of three components, and all the three components are bound by a single unit membrane. So here in this picture you can see that this is the nucleus. So this structure, reticular structure, the branch structure is the endoplasmic reticulum. And here you can see that this endoplasmic reticulum is present near the nucleus. So at one end, most of the endoplasmic reticulum, at one end they are attached to the nuclear membrane, and at the other end they are attached to the plasma membrane or cell membrane. I'll show you in the next picture. So here you can see that the structures, the three structures or the three components of the endoplasmic reticulum are the cisternae. So these are the sac-like or bag-like structures. These are called cisternae, which are usually branched. And here you can see the tubular structures, which are called tubules, and besides that the other structures are the vesicles that we will see in the next diagram. So here this is the location of endoplasmic reticulum and the structure of endoplasmic reticulum. It forms a reticuli or branched structure, so it is called reticulum. And this endoplasmic reticulum is of two types, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. So here you can see that here in the cisternae some granules are present on their surface. So these granules are nothing but another cell organelles. They are the ribosomes. So when the ribosomes are present on the surface of the endoplasmic reticulum, then they are known as rough endoplasmic reticulum. But here in this part of endoplasmic reticulum, you can see that there is no granules or no ribosomes on the surface of endoplasmic reticulum. So these endoplasmic reticulums are called smooth endoplasmic reticulum. Okay. So these are the components of endoplasmic reticulum, as I was telling you. Cisternae. So the cisternae are flattened, unbranched sac-like structures. I have already shown you. So these are the branched sac-like structures, and then they may be branched or unbranched. Then vesicles. The vesicles are oval or rounded vacuole-like elements. They are scattered in the cytoplasm. Then tubules are wider tubular branched elements mainly present near the cell membrane, and they are free from the ribosomes. Okay, remember cisternae are containing the ribosomes on their surface, but the tubules do not contain ribosomes on their surface. So the smooth endoplasmic reticulum mainly composed of the tubules, whereas the rough endoplasmic reticulum mainly composed of cisternae and vesicles. Okay. So these are the pictures of individual picture of the components of endoplasmic reticulum. So here you can see that this is nuclear membrane and this is plasma membrane. So this endoplasmic reticulum is attached to the nuclear membrane at one end, and at the other end it is attached to the plasma membrane. Okay, and these are the three components. So here you can see these are the cisternae, which are less branched or maybe unbranched. These are the vesicles, which are usually scattered in the cytoplasm. Then tubules are branched structures. Now, types of endoplasmic reticulum, I have already discussed this. But the most important thing, let me tell you, the rough endoplasmic reticulum, the short form is RER, is frequently observed in the cells which are actively involved in protein synthesis and secretion. Why? What is the reason? Because protein synthesis takes place in the ribosome, and ribosomes are attached to the surface of rough endoplasmic reticulum. That's why the rough endoplasmic reticulums are found in those cells which are actively involved in protein synthesis. Then the smooth endoplasmic reticulum do not contain the ribosomes, so they play no role in protein synthesis, but they help in synthesis of lipid. So they are present in the cells where the lipid like steroidal hormones are synthesized. So these are the some of the differences between rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes on its surface, so its appearance is rough, and in smooth endoplasmic reticulum the ribosomes are absent, so the surface is smooth. The rough endoplasmic reticulums are more stable structurally, whereas smooth endoplasmic reticulums are less stable structurally. The rough endoplasmic reticulums are mainly composed of cisternae and vesicle. This point is very important. The smooth endoplasmic reticulum mainly composed of the tubules. Rough endoplasmic reticulums are abundantly occur in cells which are actively engaged in protein synthesis. I have already told you, and the cells of liver, pancreas, goblet cells. These cells are actively engaged in protein synthesis. But the smooth endoplasmic reticulums are abundantly present in adipose tissue, interstitial cells of testes, muscles, glycogen storing liver cells, and adrenal cortex, because these cells are involved in lipid synthesis. So here is the function of a smooth endoplasmic reticulum. Since it lacks ribosome, so no role in protein synthesis, but it helps in lipid metabolism. The underlying part is very important. Lipid metabolism, carbohydrate metabolism, and it also helps in detoxification, and it is present especially abundant in mammalian liver and gonad cells. So here in this picture you can see that these pink color, the violet purple color structures are the rough endoplasmic reticulum, which are present towards the nucleus, and they are attached to the nuclear membrane. But the smooth endoplasmic reticulums are present towards the cell membrane, and these smooth endoplasmic reticulum, since they are present towards the nuclear cell membrane, so they easily remove the toxic substance from the cell through the cell membrane. Okay, so very clearly you can distinguish the rough endoplasmic reticulum and smooth endoplasmic reticulum, the structure of tubules, the components of rough and smooth endoplasmic reticulums are very much clear here. Okay, so here you can see that the smooth endoplasmic reticulum composed of tubules, and these tubules are highly branched, whereas the cisternae are less branched, or sometimes they are not branched, but they contains the ribosomes on their surface. Okay. So these smooth endoplasmic reticulums, as I have told you, they are present in the testes, ovaries, and skin, oil glands. They have large number of smooth endoplasmic reticulum because they are engaged in the synthesis of lipid. Now the function of rough endoplasmic reticulum. So, as I have told you that the rough endoplasmic reticulum contains ribosomes on its surface. Unless and until the ribosomes are attached to the surface of endoplasmic reticulum, they cannot synthesize the protein. So, the endoplasmic reticulum, their first function, foremost function is to provide surface for the ribosome to synthesize protein. Okay, so besides that, after the synthesis of the protein in the ribosome, the proteins are transported to the endoplasmic reticulum, and from the endoplasmic reticulum they are packaged in the vesicles. I have told you that one of the component of endoplasmic reticulum is vesicle. So here you can see that from the endoplasmic reticulum, the cisternae, after the synthesis of the protein, the vesicles are released, and these vesicles are packaged with proteins, and these vesicles are transported to the Golgi complex. So here you can see this is Golgi complex, another cell organelle. So this is another component of vesicles, are the component of endoplasmic reticulum, which are formed after the protein synthesis in the ribosome. They are packaging unit of the proteins. They contain the proteins, then they transport these proteins to the Golgi complex. Now the next organelle of endomembrane system is Golgi apparatus. It was discovered by Camillo Golgi in 1898. It is a reticular structure present near the nucleus, and the Golgi apparatus also consists of many flat disc-shaped sacs called cisternae, which are 0.5 micrometer to 1 micrometer in diameter, and these cisternae are stacked parallel to each other. They are stacked, they are present one above the other, and these Golgi apparatus are present in all the eukaryotic cells, but in plant cells the components of the Golgi apparatus are scattered and they are called dictyosomes. And the Golgi apparatus are absent in bacteria, blue-green algae, RBC, spermatozoa of bryophytes and pteridophytes, and sieve tubes of phloem of angiosperm. So this is important to note because they are present in all eukaryotes with some exceptions. So these exceptions are to be, you have to remember these exceptions. And dictyosome term is also important. The Golgi apparatus is scattered in plant cells, so they are known as dictyosomes. Okay. So here you can see the structure of Golgi apparatus. So here you can see the sac-like structures which are called cisternae. They are stacked. That means they are present one above the other. And each cisternae has two surface, one concave surface and other is convex surface. Very much clear here. It is the concave surface is known as C-phase. The convex surface is called transphase. So here in the Golgi complex, you can see that the transphase is present towards the endoplasmic reticulum and the C space is present away from the endoplasmic reticulum. So before describing the structure, let us see what are the components or parts of the Golgi apparatus. The parts of Golgi apparatus are cisternae. I have already told you. The cisternae has convex end, that is called forming phase, F phase or C phase. The cisternae at the concave end comprises the maturing phase, M phase or trans phase. Okay. So let me show you. So this convex phase, this is convex phase. So convex phase is formed. So these are formed by the accumulation of the vesicles of the endoplasmic reticulum. So these are the vesicles which are combined together to form the cisternae. So the phase that is present, you can say the convex phase is called transphase or it is called forming phase. But as the Golgi body attains maturity, you can see that the concave phase is present here and that is called cis phase, and it is also called the mature phase. So this is the mature end of the Golgi apparatus. Okay. So the next structure is tubule. So these tubules are formed due to fenestration. Fenestration means branching of the cisternae and form a complex network. So here you can see these cisternae are the sac-like structures. So when these sac-like structures are branched, so here you can see this is branched structure. So these are forming the tubules. Okay. The next is secretory vesicles. These are the small sized components present along the convex surface. So here let me once again show you. So these are the small vesicles. So they are packaged with proteins. They are present towards the convex end of the Golgi complex. So they are the secretory vesicles. Then Golgian vacuoles. So these are the expanded part of the cisternae which have become modified to form vacuole. So let me show you. So here you can see this portion is cisternae. Then you can see that the cisternae is expanded and it is vacuole is a large spherical structure where the processed proteins are packaged. The proteins that is released from the endoplasmic reticulum are packaged in the vesicles. Then these vesicles are combined together to form the cisternae. Then in the cisternae, the proteins are processed. Processed means the proteins are modified. In some protein, the phosphate group is attached. In some protein, sulfate group is attached. In some protein, becomes glycoprotein. Some protein becomes lipoprotein. So in this way, the protein, the pure protein, is modified or processed. Now these processed proteins are packaged in the vacuoles, and then these vacuoles are transported to their destination. So because they are packaged with various types of processed proteins, so they are dispatched to different parts of the cytoplasm, mainly for the formation of membrane of various cell organelles as well as cell membrane. And these proteins are also dispatched for the formation of the lysosome, because lysosome contains the hydrolytic enzyme which is protein. So these proteins are released from the Golgi apparatus. So here you can see in this picture, so this is nucleus at the center, then the endoplasmic reticulum, then just near the endoplasmic reticulum, the Golgi complex is present. So here you can see the vesicle, the blue color vesicles are the vesicles of the endoplasmic reticulum. Then these vesicles are, you can see the proteins are transported to the vesicles of Golgi complex. These are the secretory vesicles of Golgi complex. Then they are combined together to form the cisternae, and this cisternae processes the protein. Then these proteins are packaged in the transport vesicles or the vacuoles. And now you can see these transport vesicles here on this side and this side, on both sides you can observe these vesicles are transported to the cytoplasm for the formation of lysosome at one end, and at the other end you can see that the unnecessary proteins or some proteins are also released out of the cells, and here the vesicular proteins are also used for the formation of the plasma membrane. So if you have any doubts, then you can pause this particular slide and you can observe this diagram so that it will be clear to you, and listen to my description also. So here very clearly shown the position of the endoplasmic reticulum and the Golgi complex, how the Golgi complex and the endoplasmic reticulum functions are related. They are working in a coordinated way, even the lysosome also. So these are the functions of Golgi complex, Golgi body. They modify the proteins and lipids, because the lipid that is secreted from the smooth endoplasmic reticulums are also processed by the Golgi complex. They process the materials which are to be removed from the cells, as I have shown you that exocytosis is taking place. So those materials which are to be removed from the cell are also processed by the Golgi complex. They make and secrete mucus. They package products into vesicles for transport. So here very clearly I have shown you in the diagram how the vesicles are formed for transporting the proteins. The next endomembrane organelle is lysosome. The lysosomes are microscopic vesicular structures, and they are present near the Golgi complex usually, and they are also surrounded by a single membrane made up of lipoprotein, and these lysosomes are called the suicidal bag of the cell because they contain a digestive enzyme called hydrolytic enzyme, and this digestive enzyme digests the substances that enters into this particular cell organelle. So we'll discuss the function of lysosomes in details with the diagram. This organelle was first discovered by Belgian biochemist Christian Dedieu in 1955 in the liver cells, and he named it as pericanalicular dense bodies. And the very important to note that these are absent in prokaryotes but are present in all eukaryotic animal cells except mammalian RBC. But they are present in fungi, euglena, cotton and pea seeds, all plant cells. They are present as a spirosome. In plant cells, the lysosomes are called spirosomes. Okay, that we'll be discussing in our next session. So let me first show you the position of the lysosomes. So this is the cell. You can see the nucleus, then the cytoplasm is here. So we have discussed the endoplasmic reticulum. This is the location of endoplasmic reticulum. Then near the endoplasmic reticulum, the Golgi complex is present. Then near the Golgi complex, the lysosomes are present, which are vesicular structure. These green vesicular structures are the lysosomes. These lysosomes is the polymorphic organelle. Very, very important, children. Polymorphic. This exists in many forms. So what are the forms of lysosome? The lysosome may exist as primary lysosome, secondary lysosome, tertiary lysosome or residual body, autophagosome or autolysosome. So these are the different forms of lysosome. That's why the lysosome is called polymorphic organelle, cell organelle. Polymorphic means many morphological structures. So it exists in many more than one morphological structures. And as it performs its function, these forms are appear. So with the help of diagram, I will explain you how the primary lysosome, secondary lysosome, tertiary autophagosomes exist. Before that, let me explain what are primary lysosomes. The primary lysosomes are the newly formed lysosome, and as I have told you that the lysosome contains the hydrolytic enzyme, and this hydrolytic enzyme is a digestive enzyme. But in the primary lysosome, this enzyme is probably in an inactive state. Then the next form is secondary lysosome. So when some material need to be digested, it enters into the primary lysosome, and now the primary lysosome becomes the secondary lysosome, in which the enzyme, the digestive enzyme is active and it starts digesting the material that has entered into the lysosome. They are also called heterophagosome because it digests or engulfs and digests various substances. Then the secondary lysosome becomes the tertiary lysosome after digestion. So after digesting the material that has been entered into the secondary lysosome, the left out substance, which are called residual bodies, are remaining in the lysosome. So that form of lysosome is called tertiary lysosome or residual body. The next autophagosome or autolysosome. So this is the form of lysosome. It exists when a cell may digest its own organelle. When the lysosome digests its own organelle, worn out organelles. When an organelle becomes functionless, like mitochondria, endoplasmic reticulum, Golgi complex becomes functionless, they need to be removed from the cell. Otherwise, they will they will create toxic in the cell. So these cell organelles are digested by the lysosome. So that form of lysosome which digests its own cell organelles is called autophagosome or autolysosome. And this process of digesting the organelles of its own cell is called autophagy, and the structure is called autophagosome. Sometimes when the cell is functionless, the lysosome itself bursts, the enzyme is released, and that enzyme digests the whole cell. So that's why it is called suicidal bag of the cell. Okay. So now let us see these forms of lysosome. So as I have told you that the lysosomes are formed from the secretory vesicles of Golgi apparatus. So this is Golgi apparatus. From the Golgi apparatus, the lysosomes are produced, and these lysosomes are the primary lysosomes. This primary lysosome contains the enzymes which are inactive, but here you can see that some material has been entered into the cell, and this material needs to be digested. Then this material enters into the lysosome, the enzyme becomes active and it starts digesting that material. And this structure is called secondary lysosome or phagosome. Now after digestion, so here in this picture, third picture, you can see after digestion, the digested substances are utilized by the cell, but undigested materials remains in the lysosome, and this is now this structure, this stage of lysosome is called residual body. And sometimes, as I have told you that when some cell organelles, here I have shown you mitochondria, the mitochondria becomes worn out or functionless. Now this mitochondria needs to be removed from the cell. Then what happens? This mitochondria is engulfed by the lysosome, and this lysosome, here you can see it has engulfed the mitochondria, okay, and it digests the mitochondria. So when the form of lysosome that digests the cell organelles of its own cell, then it is called autophagic lysosome or autophagic vacuole. So here are the different forms of lysosome. That's why it is known as polymorphic organelles. And when the whole cell is damaged, when whole cell is functionless, then the lysosome, all the lysosome bursts. They release the enzyme, and that enzyme digests the whole cell. So if you will have any doubt, then you can pause this particular slide and you can listen to me, my explanation, and I can go through the diagram. So here are the function of lysosome. So heterophagy, I have already told you, when any foreign substance enters into the cell, then the lysosome digests it. So that process is called heterophagy. Autophagy means the cell organelles are digested by the lysosome. Then programmed cell death, when the cell is functionless or anything wrong happens with the cell, then there is a programmed cell death. Means, as I was telling you that the lysosome bursts, the enzyme is released, and that digests the cell and the cell dies. Then autolysis, also breaking down of the cell organelles by the enzyme of the lysosome. Then fertilization, how it helps in fertilization. You know that in the human sperm, in the head of the sperm, the acrosome is present. That acrosome contains lysosome. The hydrolytic enzyme of the lysosome present in the acrosome of sperm digest the egg membrane, hence it helps in fusion of the sperm and the egg. and helps in fertilization. So these are some of the functions of lysosome. Then the next organelle, that is the endomembrane system organelle, is vacuole. So this vacuole here in this picture, you can see that it is the structure of a plant cell, and you know that in a plant cell there is a very big central vacuole is present. A single vacuole is present in plant cell. But in an animal cell, many small vacuoles are present, and these vacuoles in the animal cell perform very specific function. So here in the plant cell, a big central vacuole is present. That vacuole is filled with the cell sap. The membrane of the vacuole is called tonoplast, very important. And here you can see that the vacuole occupies most of the part of the cell, so the nucleus is pushed to one corner of the cell. So here you can see the types of vacuoles in plants and animals. In plant cells, only one type of vacuole is present, which is filled with usually with water. Okay. So very clearly I have shown you the picture of a plant cell with the vacuole which is present at the center. The nucleus is pushed to one corner. Okay. But here in the animal cell, you can see there are different types of vacuoles are there. I have told you about the vacuoles associated with the Golgi complex. Then in unicellular organism, the contractile vacuole, the food vacuole, the water vacuoles are there. The food vacuole contains the food, and that helps in uh, digestion, absorption and assimilation of food. The water vacuole stores the water. The contractile vacuoles helps in osmoregulation and excretion. So there are different types of vacuoles present in animal cell, and they are present in more than one number. But in plant cell only one vacuole is present, that is present centrally. So here the description of different types of vacuoles are there. That is food vacuole. It is from phagocytosis fused with lysosome for digestion. Then contractile vacuole, it has in osmoregulation, that is it maintains the water balance. And when inside the body the water becomes more, then this water is collected by the contractile vacuole. The contractile vacuole comes to the surface of the body and it bursts and the water is released. Then central vacuole, it is present in the mature plants. It stores the waste, mainly water, and it also helps in maintaining the turgidity of the plant cell. So in this session, we have discussed some of the cell organelles which are forming an endomembrane system. They are working in a coordinated way. Their functions are related to each other and they help in the biosynthesis of each other. So I hope the structure and function of all the endomembrane structures are very much clear to you. And in our next session, we'll discuss the other cell organelles like mitochondria, chloroplast, and ribosomes. So these are not endomembrane system, part of the endomembrane system, because they perform very specific function individually by taking the help of the product of other cell organs. Okay, so I hope you liked this video, and please like this, give a comment, and if you have not subscribed my channel, then don't forget to subscribe to see more videos, to watch more videos. And if you want more topics related to biology, then you can write in the comment section. Don't forget to give comment. Thank you for watching the video till end. Thank you once again. Thank you very much.